Hello everybody, I'm Diego Ulisse Pizzagalli from Università della Svizzera Italiana. Thanks for the opportunity to present at the I2K conference this talk entitled Action Recognition in Microscopy, Methods and Datasets. More in detail, I will present how we can apply a technique from computer vision, which is called action recognition, to study the behavior of motile cells from microscopy videos. Then I will present how we can apply this technique to the specific case of disruptive cell death, and finally I will present the IMMUNEMAP project, which is a large dataset of microscopy videos to which everybody can contribute. Well, action recognition resembles a large number of methods which were established originally to study the behavior of humans. So these methods want to detect what a human is doing from his or her motility patterns. You can see this person is detected as running, as standing, waving, and doing other things. Well, we want to translate these techniques to understand what cells are doing. For instance, have a look at this video, which was captured using intravital two-photon microscopy in the lymph node. You can see in light blue neutrophils migrating from the inner part towards the outer part of the organ, where the influenza virus in red was trapped. You can see cells doing a variety of different things. You see some neutrophils here that are going very fast from the inner part toward the outer part. You can see other here that are forming aggregates, they are swarming, and you can find maybe some other here in the upper part that are going back and forth. Let's have a closer look at one of these neutrophils. You can see the cell is going towards this red cell, which is a macrophage. It's forming a contact and then going away. Another action. However, most of the studies employing intravital microscopy to study motility of immune cells mainly quantify a few parameters like the average speed or the directionality. This specific neutrophil has a speed of 2.5 micrometers per minute and a directionality of 0.5, meaning that it's not going straight, but it's not also following a very twisted trajectory. The goal of us is in general to extract knowledge from microscopy data, and is it possible to go beyond these numbers? Well, our hypothesis is that by using actions we can extract something more with respect to these basic parameters. So we proposed a hierarchical model to describe the content of a video. At the bottommost level we have the pixels, so these tiny dots are the pixels of one frame, the pixels of another frame, the pixels of another frame. Well, we can, for instance, find the correspondences in time between pixels via optical flow, and this can tell us a general overview on the motility of a video. We can group pixels into objects like superpixels or even cells, so I'm going towards the top, and have a higher level of abstraction. We can do this thing, for instance, using segmentation methods. And by employing tracking algorithms, we can reconstruct the tracks followed by the cells. Now we want to add another layer of complexity on top of tracks, which is action. For instance, we can group these 4 cells into an action called contact, meaning that these cells are undergoing the formation of a contact, and then we can detect maybe another actions afterwards, which is apoptosis. So we can understand that before there was a contact, and after cells underwent apoptotic cell death. And this is a way to model a biological process, like the response to an infection. For this reason, we reviewed the literature and reported the main motility patterns displayed by immune cells which were associated to a biological meaning. For instance, patrolling was reported as a strategy for immunosurveillance. For more details about these actions, please have a look at our paper. We then trained a tracklet classifier that takes as input a portion of a track and attributes to it an action. Thanks to this, we were able to spot changes in neutrophil behavior in the early response to influenza vaccination, which were not captured only by looking at the speed. Indeed, if you look at the time course of the speed, between 1 hour and 30 and 3 hours, no significant differences arise. Instead, if you look at the neutrophil actions, you can see that at 1 hour and 30, most of them were patrolling, red column, while at 3 hours, most of them were swarming. We wanted to do another step and detect cellular actions relying on deep learning without the need of tracking the cells and by looking at the cell morphology over time. For this reason, we started to train a comb LSTM architecture to detect a specific type of cell death which yields to the disruption of the membrane into pieces, like for instance apoptosis. To achieve this, we used a comb LSTM architecture, which was trained using data acquired in vivo and in silico. We gave to the classifier as negative examples migratory cells, like the one that you can see in panel I, and this is instead in panel II an apoptotic cells that fragments into pieces. We have a problem because there is currently a lack of publicly available data acquired in vivo. Therefore, we had to complement our data with other data generated in silico. So we used a digital phantom to generate a cell that breaks up into pieces or a cell that migrates resembling amoeboid migration. This provided us with the enough amount of data needed to perform the training. I want to stress the fact that for this project we did not perform any single experiment on animals, but we reused videos acquired by others in which we opportunistically found cells undergoing disruptive cell death. This is the output of our method. You see here it detected a cell undergoing disruptive cell death. I play it again, and you can see the cell that disrupts and form some debris. So when we trained the method using only data acquired in vivo with some augmentation, the learning curve was oscillating quite a lot. Instead, when we performed the first training in silico and then we used the transfer learning to retrain the network in vivo, the training was much smoother. Knowing the coordinates of dying cells is important. Have a look at this video. Here, the cell dies, and all the others go there. Okay, so we can spot and use these coordinates to quantify the response of bystander cells. For instance, by counting the number of cells in a radius, or by analyzing the directionality of the bystander cells. And you can see that following apoptosis that occurred in proximity of this red line, the other cells changed their directionality. They changed direction, so the directionality decreased, and they started to migrate towards the dying cell. You can also see here an heat map of the response, in which it's visible that the death of this cell influenced the cells up to 150 micrometers far away. Data availability is critical for the successful application of computer vision for the analysis of microscopy videos. For this reason, we started a project called IMMUNEMAP, in which we want to centralize on a cloud platform videos capturing immune cells in vivo. So researchers can upload experimental details, raw data, and benefit of a crowd service to track the cells. IMMUNEMAP can be used as a digital lab book to retrieve the experiments, for large-scale investigations by combining data from multiple laboratories, and also to support the development of novel analysis tools. Indeed, with IMMUNEMAP, it's possible this loop, where biomedical researchers upload data. This can be used as datasets for machine learning by computer scientists that can provide back novel tools that allow biomedical researchers to perform more advanced analysis. On IMMUNEMAP, we have currently more than 260 videos, more than 11,000 tracks, and 500,000 clicks done manually on the single cells that were moving. We started to publish a portion of IMMUNEMAP under the leukocyte tracking database, specifically meant to help those researchers developing tracking algorithms. Conclusions: we say that immune cells are crazily dynamic. Action recognition simplifies the interpretation of their behavior. To do this, we need data, so please deposit raw microscopy data in open repositories, like IMMUNEMAP. Thanks for your attention, and thanks to all the people that made this possible.